Thank you everybody for joining us. Sorry for the delay. My name is Jeff Chen. I'm the director of the UCLA Cannabis Research Initiative. Really excited to be here and host CannaMed here at UCLA. I actually earned my medical degree and my MBA both here at UCLA. So I'm a Bruin. Any Bruins in the audience? Yeah, we got a bunch. Great. And just a quick 30-second blurb about what is the UCLA Cannabis Research Initiative. So we started the program about a year ago, and it was through a generous gift from Terry and Jane Semmel. And our mission is to advance the scientific understanding of the impacts of cannabis on body, brain, mind, and society. And so we have 37 faculty members from UCLA who've joined the initiative. It spans over 15 different departments and 6 different schools here at UCLA. So it's an incredibly collaborative, multidisciplinary effort. And today we're here to talk about Alzheimer's disease. And by 2050, it is estimated that 1 in 2 seniors, when they pass, will suffer from some form of dementia. 1 in 2. And Alzheimer's disease is the most common form of dementia, and that's why we're here to talk about this. So I'd like to introduce our panelists. So to my right here, we have Dr. Jamie Cox. He's the Chief Medical Officer of Sundial Growers, which is a cannabis producer in Alberta, Canada. Dr. Cox works with healthcare organizations, universities, and industry partners to help them understand cannabis as a viable medical treatment. He has over 2 decades of senior healthcare leadership roles, including his role as medical director of General Motors. To my right, I also have Dr. Gary Smalls. He's a professor of psychiatry at the UCLA Semmel Institute for Neuroscience and Human Behavior. He's also the director of the UCLA Longevity Center and the director of Geriatric Psychiatry. He's developed brain imaging technology that can detect Alzheimer's years before symptoms start. He's also tested medications that can delay the onset of Alzheimer's, and he's developed healthy aging lifestyle and memory programs that are widely used throughout the US. Scientific American magazine named him the world's top 50 innovators in science and technology, and he's written the New York Times best-selling book, The Memory Bible. And finally, at the end of the table, we have Dr. David Schubert. He is a professor and co-founder of the neuroscience program at the Salk Institute. He established the first medicinal chemistry lab at the Salk Institute, and his recent work has focused on developing treatments for neurodegenerative disorders, including Alzheimer's and Parkinson's. He has developed synthetic derivatives of natural plant compounds that have much improved properties over their natural analogs. And he's starting phase 1 human studies for one of his drugs, J147, for Alzheimer's disease. He's also been conducting drug screening on various cannabinoid compounds to elucidate their neuroprotective properties in Alzheimer's disease. So please join me in a warm welcome for our panelists today. And so I think the first question to start off with is maybe let's talk about the current state of evidence and state of treatments for Alzheimer's in terms of actually the progression of the disease. What treatments are available right now? What are the benefits? What are the side effects that we're looking at?
Well, I can start with that. We have several medicines that are called symptomatic treatments, which means they help temporarily with the symptoms. They don't cure the disease. They don't stop its progression. And they've been around for several decades, so they're already directly marketed to consumers. You've probably seen ads about them on television, and we use them routinely when patients have mild, moderate, or severe Alzheimer's or other forms of dementia. There's been a lot of drug development and other treatment development in trying to come up with what's often termed a disease-modifying treatment. So that would mean that this treatment would actually get into the brain and have an impact on either stopping the disease or slowing it. One way to think about symptomatic versus disease modifying, if you had pneumonia, you would take aspirin as a symptomatic treatment, but you'd have to take an antibiotic as a disease modifying treatment. Unfortunately, more than 99% of the disease modifying treatments have failed, or actually all of them have failed thus far. We're all looking for better approaches to try to deal with this devastating illness.
Yeah, and I'd like to comment on that. Certainly, yesterday it was well demonstrated at the CME session the neuroprotective benefits of the various cannabinoids. And for us not to start looking at that seriously, I believe, is a gross disservice to potentially half y'all in the audience. Okay, because 1 and 2 of us are going to get it, so it's up to us to start making a difference. You know, the old saying, keep doing what you've got, you're going to keep getting what you've got. It's time for us to make a change. So I'm full on to the looking at the cannabinoids in the proper perspective, because, like Dr. Small said, 99.9% failure rate, it's not very good, I wouldn't say. So let's try doing something slightly different.
And maybe on that note, you mentioned disease-modifying agents. What are some of the currently used disease-modifying agents? How effective are they? What are the potential downsides?
Well, for Alzheimer's disease, there are no disease-modifying agents that are being used. There are several that are being studied and tested. And there's quite a few in development right now. Most of them have focused on trying to clear out the amyloid plaques that build up in the brain. Alois Alzheimer defined the disease in 1906 when he did a brain autopsy study on a middle-aged woman who had the clinical symptoms, and he revealed to the medical community for the first time abnormal protein deposits called neurofibrillary tangles and amyloid plaques that collect in the centers of the brain that control thinking and memory. So a lot of the drug development has been to try to clear out those plaques. That hasn't worked. At this point, they're trying to use those treatments earlier in the course of the disease. So they'll look for families where there's a genetic mutation and treat people who they know are going to get the disease as a result of the mutation to try to prevent the disease. Now, I think that's progress, and a lot of the research we're doing is trying to target people before they have dementia. This is a very slow, insidious, gradually progressive condition that our studies and other studies have shown that these abnormal protein deposits actually collect in the brain decades before people have symptoms. So I'm a big fan of trying to protect a healthy brain rather than trying to repair damage once it's extensive.
And Dr. Schubert, what are some of the mechanisms through which cannabinoids might be able to have therapeutic potential for some of the actual disease progression in Alzheimer's?
Well, I think that's a tough question, but I just sort of like go back a step because I'm a big fan of plants, and we do have synthetic drugs which we're trying to get in the clinic, and some of these are pretty well along, at least in the preclinical stage. And so if you look at the history of drug development, the majority of the drugs in the clinic today are based on plants. I mean, the chemical scaffolds are basically those from chemicals that derive from plants. So the problem with Alzheimer's, as we realized a long time ago, or so maybe 10 years ago, is that the amyloid idea was a very good idea to begin with, and where you accumulate the plaques and the tangles and whatever the initial description, but that may not be the only thing that's going on. So we took an approach that we did some library screening of a big collection of plant extracts and found some compounds that work quite well. And we developed a series of assays which mimic the old brain, what happens in the aging brain, and then we screen compounds and tried to find ones that work and then modify those so that they could make a better pharmaceutical, appropriate drug. And so the question is, in the cannabinoid field, how you would go about doing that. And I think the plant itself, as you've heard this morning, has a lot of attributes, and probably a lot of things are going on when you are exposed to this plant that you really don't understand as far as the therapeutic potential. So I think if you want to talk about specific targets, I think that may be something of a mistake, because I don't think you know what the targets should be for Alzheimer's disease. And you have a couple ideas. Obviously, the cannabinoid receptors are widely studied, but there's obviously some problems looking directly at those. So The idea has been to sort of take apart the plant and look at their 500 or so unique chemicals in the plant and screen those in assays, which we have used to identify potential therapeutics. And that's what we're going to do, hopefully. And but I think it's probably not a good idea to try to pre-select a target for this particular disease, because you're looking at genetic-based targets and looking at pre-selected targets, that sort of implies you know more about the disease and what you really do. So particularly with a lot of these neurodegenerative diseases, this pre-selected target idea is probably not good. So we like to do sort of screening assays without knowing the target and trying to find drugs that work.
And can you clarify, when you're talking about target, are you referring to a target drug to study, or are you referring to a target receptor in the body that you want to design a drug around?
Usually when you're talking about drugs, you're talking about a molecular target. And so that means one molecule the drug interacts with.
And what have you discovered as to the properties of any cannabis-derived compounds that you've done in your lab that might be relevant to neurodegenerative diseases?
Well, what's unique about our lab, I think, is the screening assays we use to pick up these activities. And they're based on, as I said, they're basing on aging, things that happen in the older brains. So age is the biggest risk factor for Alzheimer's disease, and people have known that for the last 100 years, but the drug discovery world has not really paid much attention to that, and so I think that's a big mistake. So if you look at the cannabinoids in the context of what we've learned from our own work, I think there are a number of pathways which are affected. The inflammation is a major component of most neurodegenerative diseases. Systemic inflammation contributes to that. There's something which is sort of hidden in the literature, but it's appreciated in the aging field, is this mTOR pathway, which is an anti-aging pathway, and we found that our drugs affect this very much like these anti-aging compounds, these so-called geroprotectors. And so the cannabinoids actually influence this pathway in a pretty profound way. So I think that could be a target pathway, but there's no really serious studies in the way of neurodegeneration in this pathway, this target particularly.
So you mentioned inflammation, you mentioned the mTOR pathway. There was also some work that your lab had done, I believe, with THC and amyloid buildup. Could you speak briefly on that?
So one of our drugs, which we should start clinical trial approval next week, actually, it works in part by activating one of the CB1 pathway, and I think this is an indirect activation of this pathway. So we know that these drugs clear out this amyloid, which accumulates inside neurons in very early stages of Alzheimer's disease. And we've looked at like 10 non-psychoactive components of the cannabis, and they also do very much the same thing, CBD, CBG, CBN. And one potential explanation of this, these are anti-inflammatory, they're antioxidants in some respect. And so we think that there's a structural, we've been looking at the chemistry of these molecules because we do medicinal chemistry, and we're trying to figure out if there's some structural motif which probably identifies a different type of receptor in the brain tissue that it can be engaged by these molecules. And they're clearly not working exclusively through the CB1, CB2, because we have one of our screening assays uses a nerve cell line that doesn't have these receptors. So there's something else going on. We're just trying to figure that out.
All right, thank you. And so we've been talking a lot about the underlying mechanisms that cause Alzheimer's. We just touched on some of the underlying mechanisms through which cannabinoids may have some therapeutic potential. Let's shift gears a little bit and maybe talk about, and you touched on this very briefly, Dr. Smalls, is the symptoms of Alzheimer's. And I think anecdotally there are folks who are using cannabis to manage the symptoms of Alzheimer's, and is there any grounding in science for that? What does the literature say? Maybe let's start with talking about what are some of those symptoms of Alzheimer's and how do we treat some of those symptoms right now?
So early on, when Alzheimer's strikes, people generally develop memory issues. But they also can experience mood changes and behavioral changes, social withdrawal, anxiety, depression, months or even years before the diagnosis is made. So there can be quite diverse mental symptoms that present, although memory, short-term memory, seems to be the biggest complaint. As the condition progresses, there's worsening of the memory problems, there's confusion, there are language difficulties, there's problems, of course, with paying attention, there are personality changes, there are visual spatial problems. So pretty much all areas of cognitive functioning are affected. And as you get further and further into the disease, there is more and more emergence of behavioral symptoms, aggression, psychotic symptoms, paranoia, and it can be quite difficult for caregivers to manage that. The treatments are generally the symptomatic medicines that I mentioned before, medicines like donepezil, rivastigmine, and memantine that are used to help with these symptoms. And we also try to counsel families on dealing with the difficult behaviors along the way. Another approach that we've championed and others have gotten behind this is to try to help people with their everyday lifestyle. There's been a lot of compelling research showing that genetics don't account for all the risk factors for developing dementia. Certainly age is the greatest single risk factor that was just mentioned, but people who are physically inactive, people who don't get adequate sleep, individuals who are obese in midlife, people who don't eat a healthy diet, who are stressed out chronically. These are all risk factors for developing cognitive decline, and we try to help people with that as well.
I'd like to comment. Sure. Yeah. And just to add another reality-based issue that a lot of the senior population have, especially those afflicted with early onset Alzheimer's, is the polypharmacy that we see bereft in the older population. One of my prior roles was a medical director at a large extended care facility, and the number of off-label drugs that are used in the older population is really overtly concerning to me. We're already dealing with a brain disease, as Dr. Small has elucidated, with the inflammation which leads to the Alzheimer's. Add on top of that atypical antipsychotics, benzodiazepines, neuroleptic medications, and then try to function knowing that you've also got this coexisting condition that's creeping up. And the Beers report is really underutilized in the senior population, in my opinion. And if we had a clearer focus on the pharmaceuticals that are being used in this population and started weaning these ineffective medications off, it would give us a much greater opportunity to actually impact people's lives, which is what we as physicians are responsible to do. Improve quality of life, do no harm.
So if I can, if I can just expand on that a bit more. Another common problem is people take over-the-counter medicines for sleep, or they take antihistamines that have an anticholinergic effect. Now that's important because one of the neurotransmitter systems that gets into trouble in Alzheimer's is the cholinergic system. So that will, and in fact, several of the drugs that have been developed and used for Alzheimer's are cholinergic drugs. So that's a problem. And as I mentioned, some of the behavioral problems that emerge later in the disease, which generally lead caregivers to get their family members into nursing homes and institutionalized facilities. We don't have great treatments for that. We have these antipsychotic drugs, there are black box warnings, and there are problems with that. Sometimes we use antidepressants. So there's a lot of research going on right now looking for medicines that might calm people down. Now, there have been some studies using marijuana products to treat that, and the results are a bit mixed. Sometimes they cause side effects, sometimes they seem to calm patients down.
And what are some of the, you mentioned black box warnings for some of these medicines, and kind of describe down what that is, and what are some of the risks associated with these medicines used?
So there are antipsychotics, typical and atypical antipsychotics, that can increase the risk of a stroke or increase the risk of diabetes, and so there's been a lot of concern about that. So doctors try to, even though they can be quite effective when patients are psychotic and aggressive, we're concerned about the side effects from that.
And the other issue with that is the use of atypical antipsychotics in this population is not meant to be chronic use of the pharmaceutical. If there's aggressive behavior or psychotic behavior, it's supposed to be used at that time, and then it's supposed to be weaned off. Unfortunately, I've seen in about 60% of some senior populations in extended care, they're on atypical antipsychotics until they're done.
Well, actually, there's no drug right now that's been approved to treat the psychosis associated with dementia. There are several compounds that are in development that might succeed, but we have nothing right now.
And in terms of, you mentioned some of these clinical trials of cannabis or THC, and that sometimes they're associated with side effects. What are some of the biggest concerns and risks for the use of cannabis or cannabis products in Alzheimer's patients?
Well, if patients get overly sedated, they can fall and hurt themselves, and we know that with dementia, people often have unsteady gait, so that could be compounded. They will become more withdrawn, which is a problem. A big issue I see is people, widowed individuals living alone at home, and they're completely isolated, so they can't deal with their daily activities. They're not always safe in the house. They can wander off, and if they're using cannabis, could make those symptoms even worse.
Yeah, and that's why the need for study is there. We're going to be embarking, we're fortunate enough in Canada. I am from Canada, eh? So we are fortunate enough in some provinces to have a policy on use of cannabinoids in extended care. So we're actually going to be embarking on a formalized trial, not using high THC, but rather a higher CBD, which has a more satisfactory side effect profile in the elderly population, to see if we can truly impact the anxiety and aggression that can be seen in this population.
And so maybe speaking to that, because medical cannabis has been more widely used and regulated in Canada, what is the current state? You mentioned there's certain extended care facilities that have lenient cannabis policies. What does that landscape look like right now for the use in extended care or memory care facilities in Canada?
It's really evolutionary, to be quite honest with you. The policy in the province of Saskatchewan was a guidance policy on the use of cannabinoids. Typically, cannabis is not used in extended care facilities in Canada. It's still considered to be a locked drug, the same thing as a narcotic. So it has to go through significant protocols to be even able to be used, I'll say, on the floor by the nursing staff to dispense it. That's why we're focusing on Saskatchewan first, because they've already got an accepted policy. And the goal is certainly in no way, shape, or form to create any sense of an individual being stoned. It's about trying to improve their quality of life. The ideal scenario would be to see some people wake up again. You know, from the Polypharmacy, it's bereft out there, folks, I tell you.
And so we've been talking a bit about the symptomatic management. I like to almost bring the conversation back to some of the underlying mechanisms that might target the disease. And Dr. Schubert, one question I had for you is you mentioned how you traditionally take natural compounds and then you develop analogs, so things that look similar but you modify them slightly to improve various attributes of them. And what are your thoughts as to where you want to go with your cannabinoid work? You've mentioned these various phytocannabinoids you've been working with.
Well, first off, we should define the problem, because the problem is initially, 10, 15 years ago, started working with natural products and tried to use these in a therapeutic, context of helping with Alzheimer's, Parkinson's, these types of diseases. But the problem with natural products is it's basically, from a pharmaceutical point of view, they're dead end, because nobody's going to support a clinical trial for something you go down to Walmart or someplace and buy. And so we worked for many years, and we have natural products, which we think, and other people like Greg Cole here has been working on curcumin at UCLA for years. And it's just extremely difficult to get a natural product into a clinical trial. And people in the cannabinoid field are going to, there are ways of getting around that, but the clinical trials for Alzheimer's are extremely expensive and difficult. And so somebody has to make money on it, the way the government, the United States or the world set up in order to get something into the clinic. So the only way we could get the compounds that we really thought were helpful into the clinic was by making synthetic patentable derivatives of that. And then even with that in the Alzheimer's field, it's been very difficult to get funding for the clinical trials and for the preclinical development. So that may change. There's more money in the Alzheimer's field now than there was a couple of years ago. So, but the rationale for doing the clinical medicinal chemistry is to make the drug more potent and more bioavailable, and there are problems with natural products in the sense there's some chemical issues that the medicinal chemists don't like. And so we felt in order to survive as a lab and try to move things towards the clinic, we just had to do the medicinal chemistry to get things moving. And I think there are going to be issues in the cannabinoid field of the same problem. Maybe you can organize it and learn from what people in the past working on natural products have learned, but it's going to be, I think, a little bit difficult.
So you mentioned curcumin, and we just published a study using a bioavailable form of curcumin that's manufactured by a Japanese company, and we studied people not with dementia, but people at risk for dementia. So they had what's often called mild cognitive impairment or even just normal aging. We studied them for 18 months, and we compared the compound to placebo. And curcumin is a component of turmeric, so it's basically a spice that's been around for thousands of years, and it reminds me a bit of cannabis because it ticks all the boxes in terms of effects. It's anti-inflammatory, it's antioxidant, anti-amyloid, and so forth. But we had, even though it was a relatively small study, it had a fairly robust effect on memory and attention. And so we're going to be following that up with a larger study to confirm the results. But I think this offers a model for testing cannabis products that have the right balance of ingredients, so you don't have the mental effects that might be problematic, but you have the effects on inflammation and oxidation and even possibly amyloid buildup, and you test it in people not with dementia, but people who are at risk for it earlier in the stage of their neurodegeneration.
No, I think you can go back a little bit even further than that. So there's this concept which has been around for 100 years and recently been sort of revamped by the Aging Institute at NIH and the Buck Institute up in Northern California, this idea of something called geroprotectors. And geroprotectors are, the idea is if you can find compounds which maybe extend life in model organisms and you extrapolate that to humans, and if you extend the lifespan to a little bit, you'll forestall the advent of these age-associated diseases like Alzheimer's and Parkinson. And so It turns out we've looked at a lot of the pathways involved, like with curcumin and our drugs, which are derivative of curcumin, and other the sort of known geroprotectors. For example, like caloric restriction is it's obvious what that is, and two others are metformin, which is anti-diabetic drug. It has other uses too, than rapamycin. And so it turns out that all these drugs or these compounds in caloric restriction have the same molecular pathways which lead in model organisms to life extension. And so this weekend I was looking at what's happening in the CBD, THC field, and it turns out these compounds are affecting, they send the same pathways as curcumin and some of the flavonoids and our curcumin derivatives. So I think there's this opportunity for the cannabinoid field since these drugs most of the cannabinoids are anti-inflammatory, is sort of even before you have like mild cognitive impairment or whatever in the Alzheimer's world, as some long-term geroprotector. I mean, this really hasn't been looked at as far as I know in the cannabinoid field, but I think this is a potential use for it.
No, I think eventually that may be the case. One of the challenges, though, in doing human studies, if you start very early in very young people, you might be able to have a signal on a biomarker, but to see a clinical signal is very difficult. And it isn't until you get people with what we call normal aging, which would be the precursor to MCI, or mild cognitive impairment, which would be the precursor to dementia. In normal aging, you are able to see neuropsychological tests that are quite sensitive, and you can see changes when you test compounds like curcumin. With metformin, there is a large study right now looking at lots of aging targets, and I think that makes sense. But let me also add another point. When you talk about going in earlier, we and others have studied what the brain looks like late in life in people who are heavy marijuana users early in life. And it's not a pretty picture. It turns out that you have atrophy or shrinkage of the medial temporal cortex, which is an important area of memory consolidation. So, when we talk about any kind of medication or plant or supplement, whatever it is, it's really, you got to take timing into consideration and dosage and the contents of the compound to optimize the therapeutic effects and minimize any adverse effects.
I think you brought up a great point about potential confounding factors as to timing and onset, right? There's a lot of concern about the use of cannabis with the developing brain. One area that I had a question on is there's been some work on the aged brain, this is in animal studies, that on the aged brain and the impacts of THC on memory and even neurogenesis on the aged brain. And I don't know if any of you guys have familiar with that literature or could speak to that.
I've heard very anecdotal commentary on that. And I guess we're in a unique position in Canada. I'm going to go a little bit tangent, but I'll come back to it. Cannabis is legalized in Canada. So it is. So the choice of whether to use cannabis or not is now left to the individual. We as individuals in the space of having the ability to do some research, to share our knowledge, and anecdotal is a funny word. Dr. Russo yesterday showed a slide. For those who weren't there, it was fibromyalgia, and it was 1,400 people self-reporting how they were doing on traditional pharmaceuticals, which were clinically approved, randomized, trialed, but they weren't doing very good. These people were using cannabis to treat their symptoms, and they found it superior to traditional pharmaceuticals. That's going to happen in Canada as well. So for us to be able to look at it a little bit different and look at, In Canada, we've got the reality, like I said, it's illegal. So let's make sure that we have information that we can provide to our aging population. North America, we're all getting older, we're not getting younger. So if we focus more on a holistic approach and actually identify an outcome based on how an individual is doing, I think that may be a slightly different slant on the research. The horse is already out of the barn in Canada. To be able to take a long, multi-million-dollar research-funded grant that's going to take 5 years to demonstrate that level of outcome, I don't think it's going to happen. To Dr. Schubert's point, it's very expensive to do a lot of research in the lab, whereby only 5% of any research in the lab is ever commercialized. It stays in the lab. So we have to be nimble in this new environment and perhaps think a little bit differently.
So just to reiterate, I think what Dr. Cox is saying is that you have the opportunity to almost reverse translate findings. So translation is where you go from animal studies to human studies. Here we have an opportunity to reverse translate findings where we learn from the anecdotal evidence. We don't take it for face value, but based on that, we try maybe engineer the animal studies or the clinical trials. We know what right questions to ask to actually test whether or not this is a real effect.
Yeah, and I think it's important to really demonstrate effective analytics of data. When you're looking at thousands of people with all variables not controlled, but like everyone in the room, we're all different physiologically. The unique thing, and I've seen 3,000 patients for referral for cannabis use for primary care and specialists, is it's interesting that everyone uses the same mantra when we're talking about treatment with cannabis. Go slow and gradually up-titrate to effective dose. When you're looking at some of the pharmaceuticals that are currently available, there's no ability to micro-titrate. If you got a capsule that's 300 milligrams, guess what you're going to get? 300 milligrams. So it's the education gap that we have within the physician community needs to be narrowed, but we have an opportunity as clinicians to do it correctly.
Great. And with our remaining time, I'd like to open it up for questions.
Thank you. I'd like to shed a little light on this from a physician standpoint. In Northern California, where we're using residential care facilities for the elderly as a place for cannabis use in dementia care. There are more than one regularly using cannabis on a 2-3 time daily basis. We usually start at about 10 milligrams of THC and 10 milligrams of CBD per dose. We'll go down sometimes, but usually that's a starting dose. It'll knock elderly demented patients down for a couple days, and then they get up and behave much differently with a marked reduction in their agitated behaviors. We can use the Cohen-Mansfield Agitation Index as a scale to watch these changes, and they're doing quite well. Families are reporting some improvement in cognition. Overall, the patients are happier, the families are happier, and the staff is much happier. So this is going on at this time. These are not IRB approved studies. These are people in institutions whose families want cannabis used, and under California law, we're able to do this for them. So I would hope to put some light on this as far as what we are already doing. Some of the doses have to go up very high in order to cover the needs for these agitated patients, up to 30 milligrams of THC and CBD per dose in order to control some of these very, very agitated behaviors. It's not only helping the agitated behaviors, it's helping the pain, the insomnia, and many other aspects of these elderly patients.
So could I just clarify that? So you give them a dose of 10 milligrams THC plus 10 milligrams CBD one time?
Once per dose, correct. Per
and
dose.
then how?
These are
And
whole
when's the
plant
next time?
extracts,
And then
so
you
that-
have a sustained effect after that. When do you redose them?
Usually they get a dose first thing in the morning, again sundowning at about 3 p.m. and
So it's a daily dose you use.
This is this is not a divided dose. This is a per dose. So we may go. It's 2 or 3 times daily in this patient population.
Thank you.
Yeah, and one of the elements of that, just to add on that, because of the duration that it takes for the cannabinoids to get on board when you take it orally, and the bioavailability is significantly impaired as well. As we move forward with different delivery mechanisms, you know, we talk about the mucosal oral absorption, but there's other routes as well, be it nasal sprays for the more quicker onset of action. It's going to give us an ability to titrate that aggressive behavior in a slightly different way as well.
That is true, and we are able to use oral doses as a PRN order as well in this patient population for agitation and insomnia.
Thank you. And just in the interest of time, I know there's about 6 people standing right now. Please, I just remind us, we'll keep the questions to questions and keep them brief because we only have 10 minutes and there's 6 questions we have to get through.
Okay. Hi, thank you guys for everything that you're doing. Are you familiar with Dr. Chow at University of Florida's work on Hyalolax? He's using THC, curcumin, and melatonin as a treatment for Alzheimer's. And I agree with Dr. Jeff. My mom with Alzheimer's uses cannabis as well as another friend. Same dose, roughly 11 CBD, 7 THC 3 times a day, and helps remarkably.
I'm not familiar with that work.
Check it out.
Well, there are some animal rodent studies in transgenic mice on combination of CBD and THC, and also with I think THC and CBD alone, and there's some variation in the results, and so I won't say anything specific, but in general there seems to be an improvement. It tends to agree with what we've found in simpler systems actually, and there is a pretty high-end paper in, I believe, Nature Medicine a year or so ago showing that THC, when put into the directly into the brain, has a positive effect on memory in older animals. So there's mounting scientific evidence that these compounds actually have an effect.
Also, there's a great documentary called Prescribed Grass. Some people might be familiar with it, about this population in Israel.
Yeah, but I think what you got to distinguish is, are these things working? Do you need the psychoactive component or not? And my feeling is, the way we've looked at it, you really don't for the things that we look at. And so I think the emphasis should be maybe on some other compounds, the CBD plus maybe other compounds in cannabis.
Do more research.
Correct.
We're gonna.
And again, we got 8 minutes. We got 4 more questions to go through. So please keep your questions to questions.
Yeah, I was just curious if any of you guys, especially at UCLA or in general, know of collaboration with Dr. Bredesen's work on his kind of root cause approach to Alzheimer's prevention and treatment, and using the endocannabinoid or phytocannabinoids, in collaboration with him at all.
We haven't been collaborating with him.
Okay, hi. Sorry, more of a comment too from Canada. I'm a cannabinoid physician, and specifically, I do see a lot of patients in long-term care setting. And just to your point, we are seeing some real efficacy with reducing responsive behaviors. Generally, the dosing we're seeing is 1 part THC, 4 parts CBD for most of the oils we're using. And it's really interesting because in Ontario, again, we have these anecdotal evidence, but antipsychotics without a diagnosis is a reportable outcome for the government when they do inspections of long-term care homes. So it's really, we're really utilizing it to reduce our antipsychotics in that setting, A, because it's the government's mandating it, and B, we're having success doing it. Sunnybrook there's a study going on there using nabilone, and that'll be the first randomized control trial study, and the evidence shows that they had some efficacy with nabilone, reducing responsive behaviors with dementia. So that's a new one. I don't think it's published, but that data was presented at a conference in Chicago.
Do you have a question?
Yes.
Okay, good.
This is going back to Dr. Small. I think you made a comment about the shrinkage that can occur with prolonged use of cannabis, specifically in adolescents. My question is, are there isolated studies where we know that that's occurring based on cannabis usage as opposed to maybe also alcohol playing a role in that?
So, did I see you at Woodstock? I'm trying to, no.
Potentially.
No, I mean, you bring up a point. There could be some related behaviors that could have contributed to that. And those studies didn't show shrinkage in adolescence. It showed shrinkage when those adolescents were older. So there could be confounding variables, of course.
Yes, absolutely. That's what I've come across.
Yeah.
So.
I mean, we try to control for them in the studies, but it's never 100%. Unless you're doing a prospective randomized controlled trial, it's often very difficult to tease out those confounds.
But to even catch an effect like that in any sort of a clinical trial,
I'm sorry?
To catch an effect like that in any sort of clinical trial, the follow-up would have to be quite long.
Well, the way it's done, yeah, it'd have to be quite long. The way that study was done, they recruited people who, have you been a heavy marijuana user in the past? That's how they did it.
Yeah, and the issue with that is what are they using,
Exactly.
right? Is it 2%? Is it 15%? Is it 20%? Is it contaminated? Does it have secondary pesticide? You know what I mean? That's the problem with a lot of the states that the studies rather that come out of the U.S., no disrespect intended, I used to work here. But the studies aren't legit, okay? They say, okay, people are using this. No, they don't know what they're using. So how can you have a clinical outcome based on what you don't even know what you're doing, right?
Absolutely. Are there any studies on the horizon that you guys know of that will get more specific?
Well, first of all, I wouldn't say that the studies are not legit. I would say it's a different kind of study, and
Sure.
the quality, how compelling the evidence is depends on the design. So usually when these studies are published in peer-reviewed referee journals, the referees look at all those details to try so that how they're interpreted it's done in a fair manner. So, but I would say that a randomized controlled trial, usually if it's done well, gives you more compelling evidence as in terms of cause and effect.
Thank you.
Thank you.
Hi, this one's probably more for Dr. Schubert there. I represent Dr. Raphael Meshulam, and that first intro presentation that he went through was talking about how we've been able to stabilize the acids.
Could you get closer to the mic?
Sorry. So I represent Dr. Raphael Meshulam's company, and what we've been able to do with Raphael is stabilize the acids and then create the synthetic derivatives of those. And you were mentioning that that's certainly a path that you've been taking, and I'd like to hear voices around that versus the actual natural or plant.
I'm familiar with his work, but I didn't quite understand the question.
So the question there is, you're obviously going through the synthetic option, which is something that we're pursuing.
Well, I mean, this is for us, it's very difficult to do anything but that because we don't have the facilities or the means to do any clinical trials. And considering how difficult it is to work with natural products and get any leverage as far as interest in it, it's in some sense. I mean, Alzheimer's is a particularly difficult field. So that's what we're doing. Yeah, the goal is to try to find some components in the marijuana plant that are effective, and we have to test these animal models, and if they look good, then we can modify them chemically to be able to make them into better compounds. And some of the metabolism of the cannabinoids is tricky. I mean, they break down quite readily, some of them CBD. It's so they're from the chemistry point of view, there's ways to improve these compounds, and that's what we have done, and that's sort of where we're focusing. Yes.
Yep. Have you been working with the actual acids as such, or just the molecules of the plant?
We've actually just starting this. We're just applied for an NIH grant, try to work on this. We've done some work with stuff we can buy, chemicals, cannabinoids we can purchase, but we're just starting the work.
Thanks.
Hi, my name is Dustin Sulak. I'm an osteopathic physician. I've been practicing cannabinoid medicine in Maine for the last 9 years. And one thing that we often see in cannabinoid medicine is a failure of clinical trials to be well informed by those of us on the front lines with clinical experience. So I'm really glad that you brought up this topic of kind of reverse translation. And my question to the panelists is, how can you inform yourselves on what's happening in the world of cannabinoid medicine on the front lines so that you get better efficacy and results in your trials and develop medicines that are more likely to help people? For example, in my practice, I find, like Dr. Hergenrather, that THC is extremely important and effective at sparing antipsychotics, benzodiazepines, and other Beersless medicines, and that there's typically a therapeutic window where there's a dose that reduces agitation and a higher dose that produces intoxication.
Well, the only way you can get around that, the problem is you got to do it in people. So you can go through all the toxicology studies, the dose response studies, and try initially animals to get some idea how much blood concentration you need in order to get a therapeutic effect. And then you have to find a safe dose and then go tenfold lower that, start with, with humans, see if they metabolize the drug the same way as in animals. And so now you're sort of doing this backwards. Everything is going from humans to animals. So that's the question, how that's going to work out in the long term. We'll see.
So we are out of time, but I wanted to add one thing to that, is that we actually have a roadmap, just like how the development of curcumin is almost a roadmap for potentially how we pursue cannabinoids as natural products. The FDA recent approval of Epidiolex, that was based off of overwhelming anecdotal data that precipitated drug development and interest. So there is a way to do this, there is a roadmap, and it's how can we learn through prospective observational studies, through surveys, and to help us design the clinical trials and understand the questions we need to be asking. And unfortunately, we're out of time, but I just wanted to wrap up everything that was spoken about here. Give us a round of applause for our panelists. At the end of the day, data is mixed, it's inconclusive. There's a lot of work that needs to be done. Animals aren't humans. And the last thing I'd like to add is that the use of cannabinoids for Alzheimer's disease, for both disease modification as well as symptom management, that's one of our priority areas here at the UCLA Cannabis Research Initiative. So thank you very much. Enjoy lunch. One more round of applause for our panelists.